Hello everyone, welcome to yet another session with online biology tutorials. This video onwards, we are going to study chapter number 6, Molecular Basis of Inheritance. So if you have not watched my initial videos from chapter number 1 to chapter number 5, the links are given in the description box. Please have a look. And if you haven't subscribed to my channel yet, kindly do so quite quickly. Now let's start our chapter. The basic theme of our chapter, it is going to be central dogma of biology. Now what is central dogma? You know that DNA is the hereditary material of our body and the flow of information happens from DNA to RNA and from RNA proteins are formed. So this sequential flow of information from DNA to RNA to proteins, it is known as central dogma. And here in this chapter, we are also going to study the self-replication of DNA. Means the synthesis of DNA from the existing DNA itself. Now, the process of synthesizing RNA from DNA, it is known as transcription. And the process of synthesis of proteins from RNA, it is known as translation. And here, of course, I told you that whenever DNA will be synthesized from the existing DNA, then the process will be known as self-replication. So in this chapter, we will be studying self-replication in detail, transcription in detail, and translation in detail. But prior to understanding these wide terms, we must know what is the structure of DNA. So DNA stands for deoxyribonucleic acid. Now as the name has a term in it which is nucleic acid, this is giving us the sense that it will be found in the nucleus of the cell. Now if you draw a cell, the centermost part is the nucleus and it has thread-like material called chromatin which condenses to form chromosomes. This chromatin has genes on it. So these blue structures are the genes which are the hereditary material and inside the genes DNA is found. Now, initial days when there was no technology in trend, so at that time, there was a scientist with the name Meister, and at that time, DNA was not known as DNA. It came later, right? This name came into existence a little later, but at the time of Meister, he called this DNA as to be the nuclein because it was found in the nucleus of a cell and it had acidic properties. Later, Watson and Crick, they found out the double helical structure of DNA. So, in the human body, we have DNA in the form of 2 strands and that is why we are using the term double helical DNA. Various other forms of DNA are also known like aDNA, bDNA, cDNA and zDNA, which are quite prominent. Now, in this grade, we would be studying the details of the bDNA because this was given by Watson and Crick. So, for board examination, we need to make this as the ready concept. Now, first we will see what are all the components that are found in DNA. So, in short, we have studied in grade 11 that DNA is a polynucleotidic structure. Means it's a polymer. And what is the monomer unit in it? The monomer unit in it is a nucleotide. So, we will see how a nucleotide is formed. A nucleotide is composed of a base, a sugar, and a phosphate molecule. Now, bases in DNA, they are of 2 types. They can either be purines or they could be pyrimidines. Now purines are the double ring heterocyclic structures. Now when I say heterocyclic, I want to say that other than carbon, nitrogen is also found in their structures, right? So purines are the double ring structures. If you want to draw, you can draw like this, and here nitrogen is found. So otherwise on the edges here, everywhere carbon is found, but we never show it. Whereas the purines, they are the heterocyclic structures again, but they will be single ring. Now, if we are talking about purines, then we need to talk about two bases. One is adenine and the another is guanine. We abbreviate adenine as A, guanine as G. Whereas if we are talking about pyrimidines, they are cytosine, abbreviated as C, and thymine, abbreviated as T. So overall we can say that DNA has 4 bases: adenine, guanine, cytosine and thymine. Of course, the sugar in the case of DNA, it is deoxyribose, and that is why the name of DNA is deoxyribonucleic acid. And of course the phosphate molecule, it will be represented by PO4 only. So that means if I combine one base with the sugar and the phosphate molecule, that means I am going to get one nucleation. Now let's see some important concepts that are related to the structure of DNA. On the screen you can see that I have drawn a skeletal of the pentose sugar. Why are we calling pentose sugar? Because a pentose sugar is a one that has 5 carbon in it, and deoxyribose is an example of a pentose sugar. Now what is so prominent for deoxyribose? Now if you compare DNA with RNA, DNA is always going to be more stable, right? Now anything which is more stable, that stays for a longer period of time, and that is why majority of the organisms on the earth, they have DNA as the genetic material. Now if you look at C2 position, in the case of deoxyribose, only H group is there. But if you talk about RNA, in which ribose sugar is found, at C2 position, it contains hydroxyl group, which is represented as OH. So this hydroxyl group is quite unstable because it forms free radicals in the body. So if a question comes for a short note like why DNA is more stable, then we can answer that at C2 position of the deoxyribose, we have H atom, whereas in the case of RNA, it is hydroxyl group, which is quite reactive, and that's why it is unstable. So here you can see that C1 is there. What we should know that bases, they always attach at the C1 position of the sugar with the help of glycosidic linkage, right? Again, this is important because this year onwards the board is asking for competency based questions. So they are allowed to ask such questions, and at C5 position phosphate group is attached, right? And this is by the phosphodiester bond. So that means in a nucleotide, again you can see that this is a nucleotide because we have base, we have sugar, we have phosphate also, and whenever all these 3 units are combining, we call them as to be the nucleotide. So in DNA, many such units are repeated. Now DNA is always written from 5 to 3 prime. Make this as a rule that DNA will always be written from 5 to 3 prime, and arrow always it is there, the end of the arrow it is always near 3 prime end. So we will see in the structure how it is. So this is the C1, C2, C3, C4, and this is our C5, right? So you can see here that the fifth carbon is free. That is why we can write 5 prime. And this 5 prime here means the free end of the deoxyribose sugar. Now you can see at C3 position phosphate group is attached. Now this C3 position, the phosphate group, it is attached with the C5. So this 3 to 5 and phosphodiester bonds are combining, and here you can see that the C3 position is free because it has no phosphodiester bond with any of the phosphate group. So that is why we are writing DNA from 5 to 3 prime. Means here the 5 prime end is free and here the 3 prime end is free. So this is one strand of DNA which is written from 3 to 5 prime. Now if we talk about the monomers, then we have to draw sugar and phosphate as the backbone of the DNA. Remember, whenever you are writing, you have to write S and P in alteration, because you have seen that after each sugar molecule a phosphate group is coming. That is why it is being written as SP, SP. And bases, they are always attached to sugar. So please don't attach them with a phosphate because it would be a big mistake. Now we know that the DNA is double helical. One end is the 5 prime end and the other is 3 prime end, that you have already understood. Now the other strand is also there and the polarity of that strand is opposite to the first one. So here you can see that 5 prime end is here, but here it is 3 prime. Here it is 3 prime and just close to it is the 5 prime end. So such type of arrangement makes the two strand anti-parallel to each other. So let's see how is it possible in the chemical structure. Now this is of course our C1 and this will be C2, C3, C4 and this is our C5. Of course with C1 we will be having a base, right? And at C5 position we will have a phosphate. So here also you can see that the same concept has been applied as we have seen in the initial strand. So this goes, and here you can see that our 3 prime end is free here, and here you can see that our 5 prime end is free. So this chain in the opposite direction, it is also showing the polynucleotidic structure. So we can say that the DNA, these strands, they are anti-parallel to each other. Remember, if the chemical structure of DNA is being asked, then please make the bases at C1 position only. Now we will see what is the base pairing rule in DNA. That is also very, very important. So we know that DNA has 4 bases, A, T, C and G. As a base pairing rule, we always need to keep in mind that purine will always unite with the pyrimidines. So A will always combine with T with a double bond and vice versa. Now when I say vice versa, it means T will also combine with A with a double bond and G will combine with C with a triple bond, triple hydrogen bond and vice versa. Means C will also combine with G with a triple hydrogen bond. Dear students, that's all for now. Stay tuned for the more videos. Thank you.